Let us study the prophase 1 of meiosis 1. It has 5 phases. First, leptotin, that is little, zara, zygotin, please, packaging, don't, diplotin, dance, diakinesis. Little zara, please don't dance. Leptotin, zygotin, packaging, diplotin and diakinesis. This is the mnemonic you have to use in your examination. Now we will study the first phase of prophase 1, that is leptotin. Let us see what are the different events that takes place in leptotin. In this cell you see a very well built nuclear membrane which is clear and visible, and nucleolus which is also visible in this stage, whereas the homologous chromosomes can also be seen here. For this, the white color chromosome, we will go with this brown colored chromosome, so they make a pair, they are homologous chromosome. Now for this blue chromosome, I pick this green colored chromosome and I'll just put it here, so which makes the homologous chromosome. So we have two homologous chromosomal pair. Yes, blue, green, one homologous pair, brown, white, another homologous pair. Got it? Okay. So the cell still is in diploid stage, that is 2n number of chromosomes are present in the cell. Here the compaction of the chromosome is seen, that is the chromatin becomes condensed, that is compaction, and finally you see X-shaped structure, that is chromosome, which is visible under any light microscope. So these are the events that takes place in the leptotin stage. Now let us move to the second stage, that is zygotin. Here the nuclear membrane remains as it is, and even the nucleolus also appears, that is visible, that is in the zygotin phase. Let us see what happens to the homologous chromosomes. Okay, we'll just skip this homologous chromosome now. The brown and the white both are homologous pair. In the zygotin you see synapsis. What is synapsis? Let us assume that these two homologous pair are actually having a distance. They now come very close to each other. See, this is called as synapsis. Got it? Whereas this homologous pair here, the chromosomes, they come very close to each other and they touch. This is called as synapsis. Simple? Easy? Okay. This is synapsis. The next stage is formation of a complex which is called as synaptonemal complex, because of the action of a specific proteins which is present inside the nucleus of a cell called as ribonucleoproteins. They actually allows the attachment of the chromosomes. Okay, here you could see a structure called as bivalent. See, this chromosome now formed a structure called as bivalent. Here one sister chromatid of the green chromosome and another sister chromatid of the blue chromosome. For these two, these are non-sister chromatids. Non-sister chromatids, now they are linked together. Got it? Because of a protein called as ribonucleoprotein. Now the process still continues. They come very close to each other and one chromosome now masks the other homologous chromosome. That's it. Now this structure is called as tetrad. Got it? Why? Because of the action of ribonucleoprotein. The same applicable to another homologous pair. Here let us just assume after synapsis the synaptonemal complex is seen and now bivalent becomes tetrad. See, this is now tetrad. Okay, let us see what happens in the phase packaging. This is very important phase and you need to remember the events that occurs in this stage for your competitive examination. Very important. Here we just keep this, that is tetrad. Now there is one enzyme which is called as recombinase. They now helps in a process called as crossing over. What is crossing over? We all know that chromosomes are nothing but chromatin. Yes, chromatin is nothing but DNA. DNA contains genes. Genes are responsible for your characteristics. Yes. Okay. Here whatever the genes which are present in this particular segment, okay, and the genes which are present here exactly in this particular segment of the non-sister chromatid now shared. That is, sharing of the genetic information or the piece of chromatid is taken place between two non-sister chromatids of the homologous chromosomes. The same applicable to this also. So that takes place in packeting, and the entire process is called as crossing over because of an enzyme called as recombinase, and the sharing of genetic material is called as recombinase. Got it? Okay. Recombination. Got it? The next stage after packeting is diplotin. Here you could see the blue colored chromosome and the green colored chromosome. Here the brown and the white color chromosome. But still you could see this particular blue colored chromosome, particularly the non-sister chromatid, has a small segment of green colored chromosomal chromatid. Yes, that means during the crossing over they have shared the chromatid segments because of crossing over or recombination process. Got it? Okay. The same applicable to the another homologous pair. Again, this white colored chromosome has given a piece of chromatid to the brown colored chromosome, and the same has occurred, that is vice versa. Correct? Yes. This is seen in diplotin, and the interesting thing is now after synapsis and after the formation of the synaptonormal complex and after crossing over, these two homologous chromosomes now try to separate like this. They move away from each other because their work is done. Okay, they have to be separated. During this process, you could see the non-sister chromatids looks like this, form a structure like this. It's difficult on both the sides, but here I cannot do that. So see here, if I write on it, it looks like X. So this X shaped structure, because of the separation of the homologous chromosome in the diplotin stage, is called as chiasmata. What is chiasmata? This is now chiasmata. Got it? So chiasmata is seen only in diplotin. Diplotin is the longest phase in prophase 1. Okay. Next is diakinesis. Here, let me just bring these chromosomes and keep it here. As I said, in diplotin stage, the two chromosomes, they starts to separate from each other. Now they are completely separated. See, completely separated. Okay, and the same applicable to the other homologous pair also. Okay, and the interestingly here, till the four phases. See here, the nuclear membrane and the nucleolus. Nuclear membrane, nucleolus, and even you could see all four phases contains the nuclear membrane as well as nucleolus. But in the stage or phase diakinesis, you could see the disappearance of the nuclear membranes. Yes, the nuclear membrane now starts to dissolve or disappear. At the same time, the nucleolus also starts to disappear. The next phase is metaphase 1. In that you don't even see the nuclear membrane, at the same time this nucleolus also. Hope you understood prophase 1 of meiosis or meiosis 1. After the completion of prophase 1, all 5 stages, little Zara, please don't dance, that is leptotin, zygotin, packetin, diplotin and diakinesis. Now the crossed over chromosomes, you could easily see here the segments of the chromosomes, that is I'm talking about chromatid. Okay, so they have shared their genetic information, that is between two homologous pair now. Okay, they are scattered inside a cell, and now they are entering into the next stage, that is called as metaphase 1. Let us see what happens in metaphase 1. You could see the centrioles already they have organized themselves in the opposite poles of a cell. Yes, now they releases this asterase. Now the asterase, remember I'm stressing this point, the asterase now binds to the kinetochore of this chromosome, and another asterase released from the other centriole now binds to the another kinetochore of the same chromosome. So the two kinetochores are now bound by the asterase, and which now pushes as well as pulls similar to metaphase of mitosis, similar, and they now align exactly at the equator. And the same applicable to other chromosome 2, same. So asterase now binds to two kinetochores and pushes or pulls. Finally, ultimately, they are aligned at the equator. Okay, so let us just align them. So forming a metaphase plate. Yes, this is metaphase plate. Yes, they are aligned at the equator. So metaphase plate is formed. Okay, so this is metaphase. Now we go to anaphase. We don't see anything here. Let us just bring some centrioles. We'll keep it in the opposite pole. Like this. Done. Next, I'll bring these chromosomes. Now I'll keep it. That is a transition state between the metaphase 1 and anaphase 1. That is already the chromosomes are aligned at the equator. Now they are pulled by, let me just tell you, this is very important for your competitive examination. See, this is the asterase which is now released by this particular centriole. We'll just keep it to 2 itself, so to avoid confusion. Normally centrioles releases millions of asterase from both the sides. Like this. Okay. Interestingly, the asterase which is released from this centriole will bind to the closest chromosome. Which is the closest chromosome for this asterase? This chromosome. For this chromosome. For this chromosome. For this chromosome. Got it? Okay, now the asterase will bind to only one kinetochore. This is very, very important and interesting. Okay, so they bind to only one kinetochore of the chromosome and they are now pulled towards this particular side. That is the opposite pole. Pulled. So the same applicable to another chromosome like this. Now in one particular pole you see two non-homologous chromosomes. Yes. Okay. And already you could see the genetic material which is shared. Yes, blue colored chromatid piece is still here in the green colored chromosome. And here for the brown colored chromosome, the white colored chromatid is still there. Whereas here, the another asterase will bind to the kinetochore of this chromosome and pulls it towards the other side. And say, applicable to this chromosome too. Now, can you tell me the difference between anaphase of mitosis and anaphase 1 of meiosis 1? Simple. To understand this, let us just bring this chromosome. Let us just for our understanding, I'll keep this. Let us imagine this is now anaphase of mitosis, not meiosis. Okay. Now this particular asterase will bind to this kinetochore. Okay, we'll just touch like this. And this asterase also binds to the other kinetochore. But in the case of meiosis, only one asterase will bind to only one kinetochore of the chromosome, not on both the sides. But in this case, both the sides. Now again, we'll draw this spindle fiber or this one, what you call the asterase, and now it binds to the kinetochore like this. Now both starts pulling this particular sister chromatid of one chromosome. They pull like this, and as a result, centromere divides. See, one goes to this side, other goes to this side. This happens in anaphase. That is, sister chromatids are now separated because of the separation of the centromere or because of the force produced from this asterase, pulling force. Okay, like this. But in the case of anaphase, it is entirely different. The entire chromosome moves towards the opposite pole. Why? The reason is simple. Because only one asterase binds to only one kinetochore. That's it. See, this is the stage which decides the ploidy level of the cell. Very simple. The ploidy is nothing but whether the cell in future becomes diploid or haploid. Can you tell me now? Earlier we had diploid cell in the metaphase stage. In the anaphase first stage, because of the process, you see haploid. That is, the number of chromosome is exactly half. Earlier we had 4 chromosome here, but now towards the opposite pole we have 2 chromosome which are non-homologous and 2 chromosome which are non-homologous. That's it. So it is important phase which decides the ploidy of the cell, particularly gamete, that is sperm or ovum. Done, easy. Last one is the telophase. Here in telophase, simple and easy. There is no need of explanation for telophase, because you know the telophase of mitosis, the similar process occurs here, opposite to cut cangen. What is it? Opposite to cut cangen. Again, actually you see the chromatin formation takes place. You don't see this X-shaped chromosomes. Got it? But I cannot do it again. Okay. This X-shaped structures automatically transforms into chromatin, long thread-like structure, and you see the decondensation. Okay, then untangling is now tangled, then thickening will be opposite, that is thinning of the chromatin, like that. Okay, then again centromeres now starts to move. Centromere starts to disappear in this stage, that is opposite to cut cangen. Okay, then asterase, you are not seeing any asterase here, that is also disappears. Nuclear membrane now starts to reform, so we'll have to just put the nuclear membrane. Not, this is not asterase, this is nuclear membrane. Hope you are getting my point. That's it. Okay, then the mitochondria, then the Golgi, everything starts to reform. That is in the telophase. And finally, cytokinesis. Here, remember that in mitosis, you see only one cytokinesis. Remember it, when we started with this interphase, I said G1, S, G2 phase, then prophase, metaphase, anaphase, telophase, and cytokinesis done. That is actually, the entire process takes place in a cell which undergoes mitosis. But in the case of meiosis 1 or meiosis 2 or meiosis, you see two cytokinesis. The first is after prophase 1, anaphase, or like whatever the stages, you see cytokinesis 1, where you could see a cleavage furrow that starts from this, that is the movement of the cell membrane towards the center of the cell. Okay, so this is called as nothing but called as centripetal. The force is centripetal. Got it? Whereas if it is in the plant cell, then it is centrifugal, like this. A small middle lamella originates and then it starts moving like this. That is from center to opposite side. That is actually exactly towards the periphery of the cell. Got it? So remember it. Okay, in animal cell, formation of the cleavage furrow will be centripetal, whereas in plant cell, formation of the cell plate will be centrifugal. Done, easy. Okay, so see now these chromosomes are now inside this particular cell, new cell. This is new daughter cell. Okay, and after the end of the cytokinesis 1, you see two haploid daughter cell. That's it. Next is, there is nothing here, so you have to connect this to mitosis. That's it. Okay, again it will undergo interphase, that is interphase 2 for meiosis. Then it will undergo for prophase 1, then prophase 2, metaphase 2, then anaphase 2, telophase 2, then cytokinesis 2, and ultimately you get 4 daughter cells. All are haploid. So here, whatever the points you want to write, you just copy it from mitosis and paste it to meiosis 2. Done. But for meiosis 1, you need to understand. Got it? Okay, so that's all about it. Now let me just give you some tricks or ideas or mnemonics to remember the entire meiosis 1 phases, because it will be easy for you in competitive examination. Okay, so thank you very much. Hope you understood. I believe that you understood the entire concept. How to remember prophase 1 of meiosis 1? It's very, very easy. If you just apply all this stuff in the examination, you can easily score very good marks, particularly for board examination. Normally the board examination, you get this question for your annual examination. Explain prophase 1 of meiosis 1 for 5 marks. A diagram is not at all necessary. Remember it. Diagram is not required. Okay, but you have to be familiar with the diagrams of mitosis and meiosis. That is first as well as second, because in the competitive examination they may ask some diagrammatic questions. Okay, so simple, you have to stick to NCERT diagrams. That's it. Okay, and in the competitive examination you may get some questions like synaptonemal complex occurs in which particular phase. Synaptonemal complex is seen in this particular phase, that is zygote. Okay, or like you can also use the mnemonics to arrange these important phases of prophase 1, like leptotin, zygotin, packaging, diplotin, diakinesis. That is little Zara, please don't dance. Little L, leptotin, Zara, zygotin, please, P, packeting, don't, diploting, dance, how do you pronounce dance, dance, diakinesis, dance, diakinesis, because some students get confused with the diploting or diakinesis. Some write diakinesis here in the fourth step and write diploting in the fifth step. Okay, they may lose mark. Simple, if you remember little Zara, please don't dance. Dance, the last word in the mnemonic. So diakinesis should be the last. Okay, in the Profess 1 stages. Is it? Okay. So before that, I just want to tell you this. I forgot this. ಕನ್ನಡ ಮೀಡಿಯಂ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಒಂದು ವಿಷಯ ಹೇಳಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ನಿಮಗೆ ಈ ಟಾಪಿಕ್, ಈ ಚಾಪ್ಟರ್ ಏನಿದೆ, ಸೆಲ್ ಸೈಕಲ್ ಅಂಡ್ ಸೆಲ್ ಡಿವಿಷನ್ ಕಷ್ಟ ಆಗ್ತಿದೆ ಅಂತ ಅನಿಸ್ತಾ ಇದ್ರೆ, ಅಥವಾ ಕೆಲವು ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಮೈಟೋಸಿಸ್ ಅರ್ಥ ಆಯಿತು, ಮಿಯೋಸಿಸ್ 1, ಮಿಯೋಸಿಸ್ 2 ಅರ್ಥ ಆಗ್ತಾ ಇಲ್ಲ ಅಂತ ಏನಾದರೂ ಅನಿಸ್ತಾ ಇದ್ರೆ ನೀವು ನನಗೆ ಕಾಲ್ ಮಾಡ್ಬೋದು. ನನ್ನ ನಂಬರ್ ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ. ನೀವು ಜಸ್ಟ್ ಒಂದು ರಿಂಗ್ ಮಾಡಿ ನಾನು ನಿಮ್ಮ ಜೊತೆ ಮಾತಾಡ್ತೇನೆ. ಒಂದೊಂದ್ಸಲ ನಾನು ಕಾಲ್ ರಿಸೀವ್ ಮಾಡೋಕ್ಕಾಗೋದಿದ್ರು ಅಟ್ಲೀಸ್ಟ್ ಮೆಸೇಜ್ ಮುಖಾಂತರ ಅಥವಾ ವಾಯ್ಸ್ ರೆಕಾರ್ಡ್ ಮುಖಾಂತರ ನಿಮ್ಮನ್ನು ನಾನು ಸಂಪರ್ಕಿಸ್ತೇನೆ. ಒಂದು ತಡ ಮಾಡೋಕ್ಕೆ ಹೋಗಬೇಡಿ, ಯಾಕಂದರೆ ನೀವು ಇನ್ನು ಫಸ್ಟ್ ಇಯರ್ ಪಿ ಯು ಸಿಲಿ ಇದ್ದೀರಿ, ನೆಕ್ಸ್ಟ್ ನೀವು ಸೆಕೆಂಡ್ ಇಯರ್ ಪಿ ಯು ಸಿಗೆ ಹೋಗ್ತೀರಿ ಮತ್ತು ನೀಟ್ ಎಕ್ಸಾಮ್ ಬರೀತೀರಿ, ಅವಾಗ ನಿಮಗೆ ಕಷ್ಟ ಆಗೋದು ಬೇಡ ಅಂತಷ್ಟೇ. ಸೊ ಇವಾಗಲಿಂದಲೇ ಸ್ವಲ್ಪ ತಯಾರಿ ಮಾಡ್ಕೊಳ್ಳಿ. ಏನೇ ಕಷ್ಟ ಆದರೂ ಯು ಕ್ಯಾನ್ ಕಾಂಟ್ಯಾಕ್ಟ್ ಮೀ ವಿತೌಟ್ ಎನಿ ಹೆಸಿಟೇಷನ್. ಡನ್. ಓಕೆ, ಸೊ as I was talking about lactotin, little, yes, easy. Now, in the lactotin phase, what you have to remember? Simple, 3 events. Cut, C-U-T. Here, do you remember the process of mitosis? Cut, can, gen. Yes, easy. Cut, can, gen. What is C? Condensation of the chromosome or chromatin, chromatids, whatever. If you have any confusion with chromosome, okay, write chromatin. Again, chromatin, don't worry, you write chromatin. Okay, you get marks. Condensation of the chromosome, or the other word for condensation is compaction. The compaction of the chromosome, chromatin or chromatin. Done. U is untangling of chromosome. So, untangling is done. T, thickening of the chromatids, chromosome or chromatin, whatever. Okay, so all these 3 events takes place in lactotin. So very simple, lactotin, little, then cut, condensation, untangling, thickening. Easy, done. Okay, so we go to the next one. Zygotin, again Zara is there, Z, zygotin. Here I have written 3 alphabets, S, S, B. If you have any confusion, again one more thing, if you can remember this in this fashion also. Cut, syndicate and state bank. Syndicate and state bank, SSB. Put it into a cart, C-A-R-T. Here only C-R-T is there. If you want, you can add C-A-A, that is in small letter. Okay, just put them into a cart. Okay, cart, C-R-T, that is nothing but C is crossing over. You know that what is crossing over? Sharing of the genetic material between the 2 non-sister chromatids of the homologous chromosome is the recombination. That is the another term used for crossing over. Okay. R is an enzyme which is very important for the recombination or crossing over, that is called as recombinase enzyme. Very important, you get this question. Particularly packeting is very, very important. You are not supposed to forget this. Tetrad, when during the synapses, okay, so 2 chromosomes they come very close to each other and form a structure like this. See, this is one chromosome and this is another chromosome. Both are homologous. They come and finally they bind like this. Now, how many chromosomes you are seeing? Only one from your side. Here I can see only one. Okay, so but if I see it like this, I could see 2 chromosomes, correct? The upper one and the lower one chromosome is seen. And each chromosome is containing total 2 sister chromatids. And this chromosome also containing 2 sister chromatids. Total, count it. 1, 2, 3, 4. From this side, how many sister chromatids? Sister plus another sister chromatid, that is non-sister chromatids, you could see 4, that is tetrad. This is tetrad. Okay, so that will be seen in packeting. Done. Why? These 2 homologous chromosomes they bind because of a tiny proteins present inside the nucleus called as ribonucleoprotein. Yes, in the demonstration method I have shown you how exactly the 2 homologous chromosomes align and they are held together by the ribonucleoprotein. If you have not watched that video, watch it. Okay, so done 3 events. I forgot about this. Here S is synapse. What is synapse? Two homologous chromosomes, they come very close to each other. That is synapse. Synaptonemal complex. Because of the ribonucleoproteins, now non-sister chromatids of the homologous chromosome, they are binding. That is actually they are bound together because of ribonucleoprotein. That is synaptonemal complex. Because this arrangement is bit complex. Okay, and it confuses you. Earlier you have seen two independent chromosomes, but now you see only one chromosome. But there are two chromosomes. You cannot see the other chromosome. So it's a complex structure. So it is synaptonemal complex because of the process called as synapse. Okay, next is bivalent. What is this bivalent? See this. When synapse is occurring, for example, this is one chromosome and this is the other chromosome. They come very close to each other, and this makes a structure. As if like it looks like it has got only two chromatids, so that is bivalent structure. Okay, so SSB, synapse, this is synaptonemal complex, this is bivalent. Easy. Okay, next is China, that is in a diplotin stage. So for that you need to remember don't China. So cut, state and syndicate back and put them in a card or into a card. Okay, then go to China. Go to China. What is China here? Chiasmata. Chiasmata. What is chiasmata? So after this tetrad formation, okay, so these, for example, this is the sister chromatid which shares its genetic material with the another chromatid of the non-sister chromatid, and they are now the genetic material is exchanged. You could easily see it in the demonstration class, and now they starts to separate. They move away from each other. During the moment, one arm of the non-sister chromatid and another arm of the non-sister chromatid, they are linked like this. Looks like X-shaped structure. How many X-shaped structures you see during this chiasmata formation? 2. Simple. If you want, I can write this. This is one chromosome and this is the other chromosome. See, during the separation, exactly on the sides where recombination has occurred, they form X-shaped structure. See, it looks like X. This one, I'm talking about this one. Yes, right? Simple. So that is called as chiasmata. So formation of the chiasmata takes place in diplotin. China. Next is, after going to China, drink tea, can, gen. That's it. Okay, drink tea, can, gen. That is actually the simple way. Diakinesis, dance, diakinesis. Tea. What is tea? After the formation of the chiasmata, the particular phase should get ready to enter into metaphase 1. So they have to separate. Two homologous chromosomes must separate. So during the movement of the two homologous chromosomes, you see the termination of chiasmata. So chiasmata now terminates. Done. So that is termination of the chiasmata in diakinesis. What is this can-gen? Simple. You need to remember some important phases in prophase 1 of mitosis. Done. Can. Centriole formation takes place. Or movement of the centrioles, not formation, sorry. Movement of the centrioles towards the opposite pole. A is asterase. That is actually the thing you need to write. Done. So that's all about prophase 1, all 5 stages. Easy. Little Zara, please don't dance. That is the order of phases in prophase 1. Then cut, state and syndicate bank and put them in a card and go to China and drink tea, can, gen. Just arrange it, it will be easy for you. Okay, so that's all about it from this session. And one concept is there, that is regulation of cell cycle and cell division. How exactly some other factors control the cell cycle. Okay, I think this is not the right time to explain that, because in second year PUC you have a topic that is genetics. In that I am going to explain how exactly a cell undergoes this particular cell cycle. Okay, I think you need some time to understand these chromosomes and everything, like particularly how exactly crossing over takes place, all this stuff. I can show you that two chromosomes come very close to each other and crossing over has taken place, but in molecular level something is happening inside that. Okay, you may not understand it now. So we'll stop this chapter here itself. That is the last session of this chapter. And yeah, tomorrow you will have a new chapter, that is the first chapter of your NCERT textbook and the easiest, the living world. I'm going to share the videos, tiny videos will be there, and just watch it and write down the important points. Along with that, obviously I'm going to share the notes and MCQs and everything of the chapter living world. Unfortunately, we are taking online sessions. I know that some students are facing the difficulty, even we are also helpless. If this class is taking place in the classroom, I would have interacted with you and asked you the doubts and everything. That's it. Okay, now whatever I know, I just explain it. If you have any doubts, you cannot do anything else. Only thing is you can contact. But in the regular class, there will be continuous and frequent interaction will be taking place with the teacher and students. But unfortunately, nothing is happening. Hope the college reopens soon, maybe January 15th, I guess. Okay, if that is the thing, then I'll be very happy. I don't know about you. Thank you very much.